And we now are in a position where we have to ask the question, what are those genes? This is like the black box lab that you did. Mendel and the people that followed him had worked out that there's some kind of little thingy inside organisms that's responsible for the traits that they've got, for the physical features that they have, like flower color or uh, white spots on the fur or whatever it is that we're talking about. But what exactly are they? We know that there's something, and we know that organisms have pairs of them and they come in different variant forms called alleles. But what are they? What would we, if you wanted to see a gene, what would it look like? How do they do what they do? Answering that is going to take a couple of weeks. And we can get start by going back in time to the 1600s and a London gentleman by the name of Robert Hooke. Hooke was trying out a new invention of his. He invented the compound microscope. This is not nearly as advanced or as high quality as the ones that you used in Lab 2, but the principle is basically the same. You've got several lenses, each one of which magnifies a bit, and then you put something under the objective lens. That's the little thing poking out at the bottom of this, and look at it through the eyepiece lens, which is up there at the top right. This basically works the way that yours does. Yours is a bit more sophisticated, that's all. Hooke published a book called Micrographia, which was a book of drawings of things that he'd seen, and it was amazing. It was a bestseller. Nobody ever knew what fleas looked like before. And he also looked at this. What you're looking at is a drawing of a slice of cork. You might remember that cork is the inner bark layer of a species of oak called the corkbark oak, native to Spain, if memory serves. So this is actually some plant tissue. And you'll notice that there's a lot of spaces in the plant tissue. This is one of those plant tissues that's dead when it's functional. The actual living part of the plant has died, and all that you have left are these empty spaces, which are why cork floats, by the way. They're filled with air. And those empty spaces reminded Hooke of the cells in a monastery. So he called them cells. All right, this is a term you've heard before. This should be a nice little callback to when we were talking about plant structure back around week 3, I think it was. So he was the first person to see what we now call cells, and in fact, he gave them that name based on his observations of cork. It turned out that other organisms were made of cells as well. There were other microscopists who were able to expand on what Hooke did. In 1677, a Dutch amateur scientist by the name of Antony van Leeuwenhoek was the first to see human sperm cells. Human egg cells took a little longer. We didn't spot those until 1797. Egg cells are a little bit more difficult to access. And by about 1800, people had worked out the basics of what happens when Mr. Sperm meets Mrs. Egg and makes a baby. One early idea was that either the egg or possibly the sperm, people debated this, contained tiny little duplicates of the parent. So this is a diagram right here of a little folded up baby that someone thought he saw living inside the head of a sperm cell. And when this sperm cell penetrated the egg cell, the tiny little baby would grow and grow into an embryo and then a fetus and then a baby and ultimately an adult. The cool thing is that um, if this little baby inside a sperm cell is a boy, then he's got tiny testicles that have little babies inside them. And those have tiny testicles with tiny babies inside them. And basically, the entire human species would be nested one inside the other, kind of like the world's largest set of Russian nesting dolls. It also means that, if babies grow from little tiny humans that are inside sperm cells, it's kind of severe because it means that your average male teenager kills 100 million babies a day and sometimes twice a day. Fortunately, the development of better microscopes showed that this is basically false. This is not the way that you get babies growing inside mommy. They don't grow from tiny, tiny miniature humans inside sperm cells or egg cells. Much more complicated than that. Skipping over a lot, we'll jump to 1839 with a couple of guys named Schwann and Schleiden, German scientists. Again, the only thing I would test you on is going to be last names. Don't worry about dates or anything like that. Suffice it to say that Schwann and Schleiden proposed the hypothesis that all living things were made up of cells. Some living things consist of only one cell, like many bacteria and many protists. Some are composed of 2, 4, many. You're composed of roughly 37 trillion. But the idea is that there's something fundamentally similar about all cells. You could think of cells as being a bit like bricks. Right, you can build anything from a very small building to a very big building. Anything from a little shed to the largest buildings on the UCA campus with pretty much the same kinds of bricks. Right, there's no real difference between the buildings at UCA in the shape of the bricks. There's just a difference in how you put them together. And you could say something similar for cells. There's a limited number of types of cells that can be arranged in different ways to make different kinds of bodies. They originally stated that what defined a cell was having a nucleus. It's this blobby thing with a smaller blobby thing inside. These are some of their original drawings. We now know that that's not true for prokaryotes. But we'll let that pass. They didn't know yet that prokaryotes are made of cells, but the cells are constructed in a different manner. And as Schleiden put it, though the variety in the external structure of plants is great, their internal structure is simple. This extraordinary range in form is due only to a variation in the fitting together of elementary structures which are subject to modification but are essentially identical. That is, they are cells. Everything from mosses to giant redwood trees to the welwitchias of the Namib Desert, as different as they look, are made by putting together a limited number of types of subunits in different ways and different combinations. That's what they're talking about. Schleiden and Schwann originally thought that cells might come into being by kind of crystallizing out from some sort of living fluid. There's some sort of liquid that contains the power of life in it, and like sugar crystals forming in honey or something like that, cells might kind of crystallize out. But after more work with microscopes, which had improved quite a bit, a fellow by the name of Rudolf Virchow stated that cells could only come from other cells. Every cell in your body ultimately came from the fertilized egg with which you began. And this is true for all organisms. Cells make new cells, and we're going to look today at how they do that. As Virchow put it, omnis cellula e cellula. All cells come from cells. And these cells, of course, communicate with each other on their cell phones. Yuck, 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 yuck. Right. Okay. Jump forward to the 1870s. Microscopes had gotten a lot better. That 1840s microscope is pretty simple. By about the 1870s, 1880s, microscopes were just about as good as they are now. They were starting to run up against the practical limitations of just how much you can magnify something. There's a limit to the detail you can see using visible light. And microscopes were starting to reach that limit. It was getting easier to see what is in cells. And I promise this is relevant. The artificial dye industry had taken off. A British guy had been doing some studies of a byproduct of coal, this stuff called coal tar, and he'd isolated some chemicals that are found in coal tar and made the discovery that one of them was a brilliant purple. He called it mauve, M-A-U-V-E, this purple dye. This is a dress that is colored in it from the 1860s, I think. The reason we'd actually care about this, well, first of all, it revolutionized clothing and textiles because this was the first artificial dye. Before that, you got your dyes from plants, and they were fairly expensive, and there were only a limited number of colors available. But with the discovery of mauve and then a whole bunch more after that, it became possible to color cloth in any color you could think of very inexpensively. So basically, this is what made Forever 21 possible. This is the reason why you can buy clothes in any color you like. Why this matters for biology is that some of these artificial dyes that were found turned out to be useful in looking at cells under the microscope. They increased the contrast, and some of those dyes would actually stain some things inside a cell, but not other things. So when you take a cell and you hit it with the right type of dye, or in biology, we usually call them stains. First of all, most cells are kind of transparent, and it's not easy to see what's in them. You can make them visible with the stain, and sometimes you can see things like the nucleus more clearly because the nucleus will take up the stain and the rest of the cell doesn't so much. You've already seen stained specimens when we did that microscope lab and you looked at those prepared slides. A lot of the things that you looked at aren't naturally blue and red. They've been stained to bring out detail. What the hell does this have to do with anything? In 1876, a guy named Friedrich Schneider became the first person to observe something weird about cell nuclei. Schneider was looking at the tail of salamander, I think it was, and he noticed that most of the time when you looked at a cell from a salamander tail, the nucleus is this round blob in the middle. The cell looks a little bit like a fried egg, and that's what you see on the left. But for the salamander to grow, the cells divided, And when a cell divided, the nucleus turned from a blob in the middle into a set of little stringy objects. And then the stringy objects move to the center and then pull apart. And then they fade and you get two blobby nuclei again. So that's kind of weird. What's going on here? Well, it turned out that if you zap a cell with the right type of stain, these string-like objects take up color very strongly. This is an example. This is a cell that was stained in the process of beginning to divide. And instead of a proper nucleus, you can see it has what looks like bits of yarn kind of balled up in the center. And these little stringy things that you could only see when the cell was dividing came to be called colored bodies because they were strongly colored by stains. Or more usually, we use the Greek word for colored bodies, chromosomes. This may be a term that you've heard before. And that's why we call it this, colored bodies, when you stain them. Okay, this picture was taken with a much more sophisticated type of microscope that didn't really come into widespread use until the 1970s, I think. So we've just jumped forward in time a bit. But you can see what I'm going to point out using light microscopy if you're very careful. And when people with these new, very well-made light microscopes looked as carefully as they could at dividing cells, they found that, okay, when a cell is not dividing, the nucleus just looks like a blob. When division begins, that blob kind of looks like it starts clumping into the little stringy things, the chromosomes. And at least at the beginning, each chromosome is made up of two parallel strands that are stuck together, more or less in the middle, called chromatids. So what you're looking at is a picture taken with a scanning electron microscope. This can do things that the light microscope can't, but you can see much the same thing under light microscopy anyway. That's one of the chromosomes at the beginning of division, and it's composed of two chromatids. And the chromatids join at a spot kind of in the middle, a spot called the centromere. So these are two chromatids at the start of cell division, and these are two chromosomes, and each chromosome is made of two chromatids. And then the larger one to the right is also made of two chromatids. I just haven't put them in boxes. But you can see how each chromatid is kind of this elongated, like a very long balloon. And the two chromatids join at one particular spot in the center. That's called the centromere. So those are two chromatids of the same chromosome. And they're connected at a spot called the centromere. And there we go. Okay. Now, when a cell is not dividing, you can't see individual chromosomes. If you look at a stained cell that is not in the process of division, the nucleus usually looks like a kind of granular round blob, maybe with some darker spots in it. But there's evidence that the chromosomes are present even when the cell is not dividing. When the cell's not dividing, you can't see them clearly, but they do seem to be there. One piece of evidence is that the number of chromosomes stays the same throughout all generations, and the number is typical of the species. For example, almost all human cells have exactly 46 chromosomes. And this will turn out to be very important. Something you can notice now, the chromosomes come in pairs. So if we line all of your chromosomes up from longest to shortest, we have two of the longest type of chromosomes, which is called chromosome 1, and then two of the second longest type, which are called chromosome 2, and then two of the third longest type, which is chromosome 3, and all the way down to 22 pairs of chromosomes, plus a 23rd pair that in this case is called chromosome X and chromosome Y. We'll talk later as to what those chromosomes actually do. We don't need to get ahead of ourselves just yet, but what you've got is 23 pairs of chromosomes, which means 46 chromosomes in all. And that number is different for different species. You've got 46 in most of your cells. Mice have 40. Horses have, I believe it's 64. The world record is this tiny little fern called Ophioglossum that's got 1,260. On the other hand, fruit flies only have 8. You certainly don't have to remember those numbers, and there's nothing all that interesting about them. They're just happen to be, they're just numbers. But the point is that that number stays the same throughout all generations of a cell. If I were to take one of your cells and grow it in some kind of nutrient broth and get it to where it was about to divide, when it started dividing, I could count 46 chromosomes. And then it would divide, and then the two cells might grow for a while. And when they started dividing, I would see that they had 46 chromosomes. So every one of your cells, from the fertilized egg that began you to most of the 37 trillion that you have now, have 46 chromosomes per cell. To be more precise, they've got 23 pairs. And that dance that chromosomes go through when they divide has a special name. We refer to it as mitosis. Just like if I'm, if I get up on a winter morning and I'm not wearing my slippers, mitosis cold. Right. Okay. Sorry. If anybody's curious, remember, I'll never test you on the Greek and Latin meanings of these words, but mitos happens to be ancient Greek for a thread. So mitosis basically means the things that the threads do, because chromosomes look like little pieces of thread. Now, mitosis goes through several stages, and you do have to know these and be able to recognize them very, very well. You will be tested on this considerably. I'm not making you memorize these stages because maybe kind of because I'm sadistic, but we're going to see how this all connects with genes very soon. I promise you there is a reason why I'm going through all of this. And there's a very tight link between chromosomes and genes. We'll be able to start solving our mystery of what genes are by looking at what chromosomes do. This is a cell from an embryonic whitefish. And these just happen to be good cells to study the process of mitosis. And this is a cell that's not dividing. It's just sitting there, hanging out, talking on its cell phone. and the nucleus just kind of looks grainy. Now, the chromosomes are present, they're always there. And in a couple of weeks, I'll be able to tell you what they're doing at this stage, but you can't actually make them out under a light microscope. And so a cell that's not dividing with a grainy, lumpy nucleus with chromosomes that you can't make out is said to be in interphase. So that's what a whitefish cell looks like in interphase. Once division appears, the outer skin that surrounds the nucleus, called the nuclear membrane, usually disappears. There's some critters in which it doesn't, but we don't need to worry about that. And the chromosomes start becoming visible. It almost looks like the nucleus is starting to clump. And you can see it here. Those darker blue little lumps in the middle of the cell are the chromosomes becoming visible. And when a cell is in this stage, we say that it's in prophase. Pro meaning first. So the first phase here is prophase, where the chromosomes become visible and the nuclear membrane goes away. This picture was taken by a special type of microscope called a fluorescence microscope. We actually have one of these at UCA. And if you talk to the right faculty and learn how to use it and have a good reason for it, they do teach you how to use such a thing. And I've chosen this because fluorescence microscopy is a technique for using fluorescent stains that make structures inside the cells not just visible, it makes them look like they've lit up and glowed. So what you're looking at here is a cell in prophase. And what they've done is the chromosomes have been stained with a fluorescent blue dye. So that blue kind of lumpy stuff in the center of the picture, that's the chromosomes. In green, what you have is a couple of tiny organelles called centrosomes that start spinning a set of fibers called the mitotic spindle. And these things that look like, I don't know, like green bunches of hair extending towards each other, or I don't know, green octopus or something like that, that's the mitotic spindle. This is not easy to see with ordinary microscopy. As you'll see, you can make it out just a little bit, but it's much more clear if you use fluorescence microscopy. The reason this matters is the fibers of the mitotic spindle extending from the centrosomes, that acts as the motor. That's what actually pulls the chromosomes around in the movements that they're going to make. And the first movement that they make is the chromosomes line up in the center. So here we've got a cell. You can see the chromosomes are there in the middle. If you look at the lower left and the upper right, you can see a pair of little round blue specks. That's the centrosomes. And you can just vaguely kind of make out the spindle fibers extending from the centrosomes. It's not really clear, but the chromosomes have all lined up in the middle. The line that they form is an imaginary structure called the metaphase plate. There's no physical metaphase plate. The metaphase plate is just the plane that the chromosomes line up on. And those spindle fibers are what does the moving around. And here's a fluorescent picture of what this looks like. Again, the chromosomes themselves have been stained fluorescent blue, and you can see them in the center as kind of a blob. And then the spindle fibers are these fluorescent green kind of star-like structures that are attaching to the chromosomes. Oh, I've got a request. Can I go back one slide? Whoops, sorry, did not mean to do that. So at this stage called metaphase, right? Prophase was first. Now we have metaphase, as in I never met a phase I didn't like. The chromosomes line up in the center along this imaginary plane called the metaphase plate. And what's moving them around is those spindle fibers. The spindle fibers that are created by these little speck-like structures called centrosomes. Do you mind if I go on? Everybody got that? Okay, I'm checking the chat. Shall I continue? Chat. Okay, great. So that's what this looks like under a light microscope. This is what it looks like under a fluorescent microscope, which shows you those spindle fibers much more clearly. Now what? Remember that at the beginning of cell division, every chromosome consists of 2 chromatids, and the chromatids are stuck together at one spot, usually but not always in the middle of each one, called the centromere. In this third stage called anaphase, the chromosomes break apart at the centromere and the separate chromatids move to opposite sides of the cell. There on the left and the right, you can see those little very small blue specks. That's the centrosomes. And those 2 dark blue masses that you see are masses of chromatids that are being pulled to opposite sides. So every chromosome breaks apart at the centromeres, and each of the separate chromatids is pulled to opposite sides of the cell. I will post not just the video for these slides, but I'll also post, there's some really good time-lapse photography, some time-lapse movies that show you what this looks like as it's happening. The actual process of cell division takes, it's roughly an hour. It varies a bit. An hour is a decent estimate. That's how long the whole thing will take. But using time-lapse photography, you can see all of these stages moving from one to the next over the course of just a few minutes of footage. So I'm going to have you guys watch those. Here's what it looks like under a fluorescent microscope. There you can see the blue stuff. That's 2 masses of chromatids being pulled to opposite ends of the cell by the spindle fibers. And then finally, telophase. Telos happens to be Greek for the goal. Telophase is the phase that was the goal of the whole thing. These separated chromatids now cluster into new nuclei, new nuclear membranes form, and the chromosomes start fading out. You lose the ability to see them as individual things. And the nuclei go back to looking grainy, except now you've got 2 nuclei where you used to have 1. That's telophase. Usually the cell itself divides at this time, and what you can see has happened here is that what was one cell has kind of pinched in 2. Now you have 2 cells that are stuck together, making that shape that looks kind of like a number 8 on its side. When this entire cell splits, we call this cytokinesis. Cyto means a cell. I think it was originally Greek for a box, but it's what we use to mean cell. And kinesis means movement, right? Some of you might be interested in the Department of Kinesiology. That's the study of how people move. Incidentally, usually telophase and cytokinesis happen at the same time. There are cases where it doesn't. And if telophase happens but cytokinesis does not, you end up with a cell with two nuclei. There are organisms where cell division will take place many times, but cytokinesis doesn't, and you end up with a giant cell with thousands of nuclei. There's a type of organism called slime molds that do that. But usually, every case I can think of in humans, telophase and cytokinesis happen together, and telophase is the end of the division of the nucleus. Cytokinesis divides the entire cell. And there's what the spindle fibers look like at telophase. The chromosomes clump together to form new nuclei, and then the spindle fibers disappear. In plant cells, by the way, cytokinesis looks a little different because a plant cell can't just pinch apart. If we go back to this picture of cytokinesis in the fish embryo, cytokinesis is a little bit like, you could do this with a water balloon, right? You ever play with a water-filled balloon? And you can just use your fingers and pinch it into two separate little bubbles. Or maybe if you've ever had balloon animals made for you, you see how one balloon can be twisted to make two separate bubbles. That's a little bit like how cytokinesis works. You can't do this in plant cells because of the cell wall. Plant cells can't just simply pinch apart. Cytokinesis in plant cells looks a little bit like remodeling a house. If you have a big room and you decide you want two, you can just build a wall down the middle of the big room. And this is what plant cells do. They'll build a new cell wall across the cell. So this is a plant cell in telophase. You can see the two new nuclei are starting to form, these lumps of red. And then right across the middle, you can see, that little shadowy line. That is the new cell wall that's getting built. And eventually it will partition this plant cell into two new cells. Okay, that's kind of a lot, but I've got just a couple more to hit. When mitosis starts, every chromosome consists of two chromatids. At the end, each chromosome breaks apart, and each chromosome in telophase consists of only one chromatid because those two chromatids separated at anaphase. But if you were to watch a cell divide and then watch the daughter cells when they divided again, every daughter cell would start with chromosomes consisting of two chromatids. So somewhere where we can't see it during interphase, each chromosome somehow makes a new chromatid. How it does this, we're going to have to wait until we've looked into what chromosomes are actually made of. They're made of this stuff called DNA that you've probably heard of, but let's not get ahead of ourselves right now. But even when people didn't know about that, they knew that interphase, there must be something going on because a cell goes from telophase to interphase with every chromosome consisting of one chromatid and comes out of interphase with every chromosome consisting of two. So somewhere in the middle of interphase, every chromosome makes a new chromatid. And what we do now is divide a cell's life into what we call the cell cycle. Mitosis is what we've seen. It consists of prophase, metaphase, anaphase, telophase. I'll go back in just a second. And then in interphase, there's a beginning phase of interphase where the cell just grows, and we call that G1. There's a time period where the cell builds new chromatids. That's called the S phase for synthesis. And then there's a stage where the cell grows some more and gets ready for mitosis, and we call this G2. Exactly how long the cell cycle lasts depends on the type of cell. There are some that might be in G2 for many hours. There's some where G2 is almost nothing. It's very dependent on the type of cell, but this is why we now divide interphase into the G1 phase, the S phase, and the G2. Okay, somebody asked me to go back to this slide. It is 10:50 now, and if some of you need to leave about now, so I will go ahead and stop the, whoops. I'll go ahead and stop the recording, but I'll leave this up for a minute if people need me to.